Miniature medical devices, or MMDs, offer a way to navigate the body's tortuous vascular pathways and deliver targeted therapies directly to diseased tissue. But steering these microscale tools deep inside the human body requires continuous, non-invasive imaging to monitor their exact location. The clinical standard for this is X-ray fluoroscopy. It provides near real-time resolution and can penetrate deep into biological tissue. However, an X-ray is a two-dimensional projection. It compresses layers of three-dimensional anatomical clutter into a single, flattened plane. When these tiny devices pass beneath dense anatomical structures, particularly bone, the physical mass of the tissue heavily occludes them from view. The resulting clinical video feed is highly dynamic, obscured by sensor noise, and suffers from extremely low contrast. Requiring a human operator to manually search for and track these flickering sub-millimeter objects frame by frame leads to rapid cognitive fatigue and operational errors. Real-time perception has replaced mechanical actuation as the primary technical hurdle for deploying MMDs in vivo. Deep learning computer vision models provide an automated method to track these devices, replacing manual human observation. Training any highly accurate computer vision network requires massive, accurately annotated datasets so the algorithm can learn the visual signature of its target. This creates a data scarcity trap specific to in vivo microrobotics. Capturing thousands of hours of MMDs navigating inside actual human patients requires specialized equipment and is heavily restricted by medical privacy regulations. Even when researchers do collect real clinical data, forcing humans to manually annotate microscopic, low-contrast pixels frame by frame introduces severe inconsistencies. To bypass this bottleneck, researchers developed the MicroSyn-X framework. It is an end-to-end synthetic pipeline that generates entirely artificial, automatically labeled, and heavily randomized X-ray training data. Solving the perception problem requires mathematically generating the surgical environment from scratch, rather than attempting to film it. Training MicroSyn-X requires generating a realistic physiological background. Phase 1 uses a pix-to-pix stable diffusion model, an image-to-image translation network converting a simplified input map into a detailed output. Users provide structural conditioning by feeding it precisely drawn anatomical masks. The system uses 3 distinct mask channels for control, one for general tissue areas, one for metallic surgical devices, and one for lumens, which are the fluid-filled biological tubes inside the body. Text prompts are dynamically assembled, like porcine brain within the skull, to guide the model in generating specific tissue textures and densities. This hybrid approach creates anatomically realistic variations in organ geometry and X-ray density, all without ever scanning a real patient. Strictly controlling this synthetic anatomical canvas provides the exact foundational variability needed for robust model training while circumventing patient privacy constraints entirely. Phase 2 integrates miniature medical devices into this newly generated synthetic tissue. Simply pasting a picture of a microrobot onto a background layer fails to train the perception model. To correct this, the framework uses a physics-informed add-weighted strategy designed specifically to replicate the unique mechanism of X-ray imaging. X-ray imaging relies on differential attenuation. As beams pass through the body, denser materials absorb more radiation, modulating the visual signals of the tissue they overlap. The fusion algorithm mathematically sums the pixel values of both layers, perfectly mimicking this two-dimensional attenuation. Because the computer algorithm placed the medical device into the image, it already knows its exact pixel coordinates. This precise coordinate data automatically generates pixel-perfect bounding boxes and classification labels for the entire training set. Physics-informed fusion creates mathematically accurate ground truth training data, completely eliminating the need for laborious human annotation. Phase 3 prepares the network for real-world clinical chaos. This scatterplot illustrates domain randomization, comparing a dense cluster of real-world data to a massive cloud of synthetic data. The system intentionally scrambles environmental parameters, constantly altering mask shapes, tissue gradients, and injecting artificial noise. It applies this same extreme randomization to the devices themselves, rapidly varying their quantity, geometric shape, spatial position, and visibility thresholds. Extreme variance prevents the neural network from relying on spurious background textures, forcing it to identify invariant geometric contours. Overfitting to limited clinical data creates a fragile distribution, whereas domain randomization builds a robust feature distribution, accounting for nearly any visual anomaly. The model trained exclusively on synthetic data outperforms models trained on real data when tested in low-contrast, high-noise clinical environments. For extreme medical edge cases, systematically randomizing an artificial reality produces a more resilient perception model than reality itself. With the software training pipeline complete, physical hardware is required to close the control loop. The external setup relies on a 7-degree of freedom robotic arm wielding a rotating permanent magnet positioned directly over the patient. The trained computer vision model acts as a continuous visual feedback loop, translating the live X-ray perception into sub-millimeter robotic deployment. However, dynamic locomotion under dense bone will cause dropped frames and complete visual occlusions. Signal loss is inevitable. To maintain tracking, the system deploys software redundancies, specifically an adaptive Kalman filter. This is an algorithm that estimates the state of a dynamic system from a series of incomplete or noisy measurements. Combined with strict geometric consistency checks, this filter mathematically interpolates missing positional data during complete visual blackouts, predicting where the device is headed. This creates a system of supervised autonomy. Human operators issue high-level commands, while the algorithm autonomously executes the micro-navigation and tracking. True robotic autonomy in vivo requires a perception model that can predictively bridge the gaps in its own vision. The ultimate physical stress test for this tracking pipeline is the deployment of highly deformable liquid miniature medical devices. These devices are made of ferrofluids, shape-adaptable liquid metals capable of navigating confined, uneven anatomical spaces. The robotic arm's magnetic force gradients physically pull the droplets forward, while shifting the magnetic field orientation induces extreme shape deformation to fit through tight passages. This creates a tracking nightmare for computer vision. The target object constantly changes its morphology while darting through dense anatomical clutter. Even so, the vision model successfully tracks ferrofluid swarms as they dynamically split into smaller units and recombine. This continuous tracking is maintained even beneath 25 millimeters of solid bone occlusion and severe physiological noise. MicroSyn-X replaces a laborious and error-prone manual process with an automated, precise clinical intervention. Solving perception in complex medical environments ultimately requires computationally generated data that accurately models the physics of both the imaging modality and the anatomical domain.